Hello friends, in this video we are going to discuss about chromosome organization. Under this we will discuss about histone proteins, non-histone proteins, then we will see the overview of levels of packing or packaging of DNA into chromosome. Then we will discuss these levels of packing in detail, giving emphasis on the structure of nucleosome core. So stay tuned. We know that DNA is the genetic or hereditary material that is present inside the nucleus of a cell, if we talk about eukaryotic cell. So, it is required to compact the length of DNA by a remarkable amount in order to fit it inside nucleus. So, there is a need to compact DNA so that it can fit in the nucleus. Hence, eukaryotic DNA is organized into chromosome and DNA is present in the nucleus in the form of chromosome. In order to fit a DNA molecule into its chromosome, a highly organized packaging system is needed. Here we are going to discuss about this packaging of DNA into chromosome. During the packaging, DNA binds to many different cellular proteins. Amount of protein to which DNA binds is twice that of the DNA itself. Also, DNA along with protein is known as chromatin. Hence, chromatin is what? Nucleoprotein complex. That is DNA plus proteins. It is a dynamic structure. Why dynamic structure? Because it is capable of changing its shape and structure during the life of a cell. Chromatin is condensed chromosome at metaphase stage and it is very diffuse structure in interphase. So, we can say that it is a dynamic structure. Chromatins are made up of DNA and two types of proteins. First is histone protein and second is non-histone protein. Now, the histone proteins, these are the most abundant proteins. These are small basic proteins and have net positive charge. This positive charge is due to the presence of high percentage of basic amino acids like arginine and lysine. H1, H2A, H3, H2B, H4 are 5 primary histones found in eukaryotic chromosomes. Among the 5 histone proteins, 4 histone proteins H2A, H2B, H3 and H4, their amino acid sequences are highly conserved. So, they have the role in organizing the DNA in chromosome of all eukaryotes. So, in all eukaryotes, these 4 types of the histone proteins have a role in organizing the DNA in the chromosome. Also, they share a similar tertiary structure. Hence, these are ultimately evolutionarily related or these are related in the course of evolution. But if we talk about H1 histone, these are not conserved, but these show variation between species. It is not the part of nucleosome. It is linker histone associated with linker DNA and it lies at the point where DNA enters and leaves the nucleosome. Also, it functions as a clamp around the nucleosome. Hence, it restricts the DNA into its place. So, the nucleosome along with H1 histone protein is known as chromatosome. So, this is the nucleosome which is the octamer of 4 histone proteins H2A, H2B, H3 and H4. Hence, this H1 histone is not part of nucleosome. It is attached to the or it binds to the linker DNA. Hence, it is known as linker histone. It binds to the region where the DNA enters and leaves the nucleosome or octamer. So, H1 histone is not the part of nucleosome and nucleosome along with H1 histone protein is known as chromatosome. The detailed structure of the nucleosome, chromatosome and the further level of packaging of DNA we will discuss in detail in the next part of this video. Second is non-histone proteins. All proteins associated with DNA except histones are non-histone proteins. Example, proteins binding to DNA during replication, transcription, recombination, repair, etc. These are mainly negatively charged, hence bind to positively charged histone proteins. These differ in number and type from organism to organism, from cell to cell in same organism, and sometimes these differ in the same cell. Now let us see the overview of the levels of packing or packaging of DNA. This is the double helix of DNA, which has the width of 2 nanometer. In the next level of packaging, DNA is complex with histones, and these histones form nucleosomes. Each nucleosome is an octamer of 4 different types of histones, and DNA is wrapped around these nucleosomes. H1 histone protein binds to the linker DNA. DNA that is present between the two nucleosomes are known as linker DNA. To this H1 histone binds, and then nucleosomes along with H1 histone is known as chromatosome. So this type of chromatin appears as beads on a string. It has width of 11 nanometer. Further, this beads on string structure then coils helically, which has the width of 30 nanometer, and each turn of the helix consists of 6 nucleosomes. These are known as the 30 nanometer fiber. These are also known as solenoid fiber. In the next level, this 30 nanometer fiber forms loop, which has the width of 300 nanometer. Now, this 300 nanometer fibers are further compressed and folded and becomes more condensed, having the width of 700 nanometer. This 700 nanometer fiber is further tightly coiled. Now the width is 1400 nanometer. And the chromosome that appears during the metaphase is due to this level of packaging. So this is the metaphase chromosome and this is the centromere. Now let us see the above discussed levels of packaging of DNA into chromosome in detail. First level of packaging, which results in the formation of nucleosome. Nucleosome was described by R. Kornberg in 1974. It is the simplest, basic, structural and fundamental unit of chromatin. Proteins that are responsible for this organization are histone proteins. 4 different types of the histone proteins, H3, H4, H2A and H2B, are involved in the formation of nucleosome, that is octamer. So the histones are assembled as octamer. First of all, H3 and H4 histone protein form heterodimer. 2 heterodimer then make a tetramer. This tetramer is flanked by 2 heterodimers of H2A and H2B, and this create histone octamer, that is nucleosome. So this is one heterodimer of H3H4 and this is the second heterodimer of H3 and H4. These 2 heterodimers then form a tetramer. This tetramer is then flanked by 2 heterodimers of H2A and H2B histone, and this results in histone octamer, also known as nucleosome. Now wrapping of DNA on nucleosome. Nucleosome is wrapped or surrounded by DNA of 146 base pair length. This coiling of DNA around nucleosome is left-handed coil and it wraps nucleosome 1.67 times. Therefore, DNA is compacted by 6 folds and nucleosome resemble as beads on a DNA strand. Hence, chromatin at this level is referred as beads on a string or thread. So this is the histone octamer or nucleosome, and 146 base pair of DNA surrounds it, or it is wrapped by DNA of 146 base pair length. So this is the DNA double helix, which has the width of 2 nanometer. When the histone proteins organize as octamer and DNA is wrapped around it, it has the width of 11 nanometer and it appears as beads on a string. These beads are actually nucleosomes and string is the DNA. Now the second level of packaging of DNA, it is known as 30 nanometer chromatin fiber. It is brought about by H1 histone. H1 histone binds to linker DNA at one end of nucleosome and to the middle of DNA segment that is around nucleosome core. So nucleosome along with H1 histone is known as chromatosome. Hence, H1 histone is not the part of nucleosome core. H1 histone we have discussed in the previous video also. It functions as a clamp. It restricts the DNA into its position. Each chromatosome encloses 167 base pair of DNA, 146 or 147 base pair that is wrapped around nucleosome, and 20 base pair to which H1 histone binds or that binds with H1 histone protein. This is the nucleosome core, which is an octamer of 4 different types of histones: H3, H4, H2A and H2B. DNA is wrapped around this nucleosome core. H1 histone protein is not the part of nucleosome, but it binds to linker DNA at one end of nucleosome and to the middle of the DNA segment around nucleosome core. Nucleosome plus H1 histone protein is known as chromatosome. Nucleosomes or chromatosomes are present at regular intervals. Strands of DNA between them, that is nucleosomes or chromatosomes, are known as linker DNA. Nucleosomes compact into a structure having 30 nanometer diameter. Hence, it is known as 30 nanometer fiber. There are 2 possible models of compaction at this level. First is the solenoid model. Linear DNA with nucleosomes are coiled into left-handed helix. 6 nucleosomes are present per turn. There is central cavity that is stabilized by linker DNA. Helix model, this arrangement is in a zigzag ribbon that twists or supercoils. Solenoid model is the most popular or most accepted model. It compacts DNA 40 folds or 40 times. So what we have seen that nucleosomes or chromatosomes are present at regular intervals, and the DNA between the 2 nucleosomes or chromatosomes are known as linker DNA. Linear DNA along with nucleosome, which is known as the beads on a string, is coiled into left-handed helix known as solenoid, and the diameter or the width of this structure is 30 nanometer. This is known as 30 nanometer chromatin fiber. Each turn of the helix consists of 6 nucleosomes, or there are 6 nucleosomes per turn of the helix. Next is third level of organization, and this is caused by non-histone proteins. 30 nanometer fiber form loops. Each loop is anchored at its base by proteins in the near scaffold. This scaffold is made up of non-histone proteins. This is 30 nanometer chromatin fiber. It forms loop. The length of the loop is 300 nanometer. The base of the loop is anchored to the scaffold. This scaffold is made up of non-histone protein. This non-histone proteins we have already discussed in first part of this video. You can refer it. The link is given in the description box. Each loop has 20 to 100 kilobase pair of DNA. That is, each loop is made up of 20 to 100 kilobase pair of DNA and length of each loop is 300 nanometer. Number of nucleosomes per loop is 180 to 300. That is, there are 180 to 300 nucleosomes present per loop. DNA has scaffold associated regions that bind to non-histone proteins to form loop. So there is certain stretch of DNA known as scaffold associated regions that binds to the non-histone proteins, which results in the formation of loop. Next is fourth level of organization. 300 nanometer loops are compressed and folded to result a 250 nanometer wide fiber. The 300 nanometer loop is compressed and folded and it results in 250 nanometer wide fiber. This is 250 nanometer wide fiber. In the fifth level of organization, tight coiling of this 250 nanometer fiber takes place. This yields chromatid of a chromosome having the width of 700 nanometer. And this chromatid is visible in metaphase chromosome. 300 nanometer loops are compressed and folded and gives 250 nanometer wide fiber. Further, 250 nanometer wide fibers are tightly coiled and gives chromatid of a chromosome that is visible in metaphase. Width of this chromatid is 700 nanometer. Also, the entire metaphase chromosome has two chromatids. So the total width is 1400 nanometer. This is sixth level of organization. Chromosome visible in metaphase or the metaphase chromosome has two chromatids. Hence, total width is 1400 nanometer. This is the last level of organization of DNA into chromosome. In this way, DNA is packed into chromosome that is present in the nucleus of eukaryotic cells. So what we can conclude that ultimately chromosome that is produced is 10,000 times shorter and 400 times thicker than naked DNA. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.